So in a chemical synapse, we're going to see an electrical signal converted to a chemical signal. It's going to interact with the postsynaptic membrane, and it's going to be reconverted back into an electrical signal. From electrical to chemical to electrical. Now we have to think about how it gets converted back into an electrical signal. How does that neurotransmitter chemical bind to a receptor and generate an action potential? We know how action potentials are generated. We know that sodium channels have to open up to get things started. We need a depolarization to occur if we want to generate an action potential. So this is going to involve sodium channels. Let's talk about how this happens. On the postsynaptic membrane, there are two types of neurotransmitter receptors. They get these unfortunate names that are difficult to say and difficult to spell. There are ionotropic receptors and there are metabotropic receptors. The ionotropic receptor is what we call a direct acting receptor, and the metabotropic is what we call an indirect acting receptor. Now, what do I mean by direct and indirect? By direct, I mean that the receptor for the neurotransmitter is actually an ion channel. When the neurotransmitter, and I have it abbreviated as NT here, when the neurotransmitter binds to the ionotropic type of receptor, it causes it to change shape. And that shape change opens up the ion channel. The receptor is an ion channel. Ions can pass through the receptor as soon as the neurotransmitter binds. Now, sometimes ionotropic receptors are sodium channels. So, when the neurotransmitter binds to the ionotropic receptor on the postsynaptic membrane, sodium will rush through into the postsynaptic cell and we'll have depolarization. We will regenerate action potentials. We have recreated the electrical signal in the postsynaptic cell. But the nervous system is a little more fine-tuned than that. Not all ionotropic receptors are sodium channels. Some are potassium channels. So you can imagine if a neurotransmitter binds to an ionotropic receptor that's a potassium channel, and it causes the potassium channel to open, that means potassium will flow out of the postsynaptic cell. We will get a hyperpolarization. Now it's even harder to generate an action potential. In other words, on the postsynaptic cell, depending on the receptor and depending on the neurotransmitter, we're either going to stimulate or inhibit action potentials in that second cell. And the nervous system is capable of both. So we have a very fine-tuned response here. With a chemical synapse and neurotransmitters, we can see either stimulation of action potentials in the postsynaptic cell or inhibition of action potentials in the postsynaptic cell. Now, when it comes to the metabotropic receptor, the indirect acting receptor, all we're talking about is one additional step. We're still going to see ion channels opened up. Sometimes those ion channels will stimulate action potentials, sometimes they will inhibit action potentials, but there's one more step involved. A metabotropic receptor is not actually an ion channel. So when the neurotransmitter binds to it, it changes shape and it triggers another messenger molecule. That second messenger, as we call it, is going to travel to a nearby ion channel and open it up. So there's just one more step. In an ionotropic receptor, the neurotransmitter binds to the receptor and the receptor is an ion channel. In a metabotropic receptor, the neurotransmitter binds to it. It activates a second molecule that goes to a nearby ion channel and opens it up. You can imagine that because there's an extra step involved with a metabotropic receptor, there's just a little more delay. It's a slightly slower response than with an ionotropic receptor. Now we're still talking about fractions of a millisecond here, but there is a slightly slower response with metabotropic receptors. On this slide, we're looking at drawings of the two types of receptors in the postsynaptic cell. We're looking at, on the left, an ionotropic receptor, and on the right, we're looking at a metabotropic receptor. So this blue area right here represents the synaptic cleft. There would be a presynaptic neuron over here. Then there's the cleft, and then there's the postsynaptic cell. Maybe it's a neuron, maybe it's a muscle fiber, maybe it's a gland. It's some second cell that this neuron is communicating with. So the signal comes down the presynaptic terminal branch of the axon. We release this neurotransmitter, shown as a blue ball. It travels across the cleft and it binds to this protein that's sitting in the postsynaptic membrane. It binds to it and it causes it to change shape. The shape change, which we're looking at over here, simply opens up a channel in this protein. This protein is an ion channel. It's normally closed. But when the neurotransmitter binds to it, it opens up. Now, this could be a sodium channel, this could be a potassium channel. We're going to see ion movement through this channel, whatever it happens to be. This is the direct acting or ionotropic receptor type. Over here we have the metabotropic receptor type. So in the synaptic cleft, the neurotransmitter, shown in orange, is going to bind to the receptor and cause it to change shape. But this receptor is not an ion channel. When this receptor changes shape, it activates some other molecule. That's what they're showing us in yellow. That molecule we call a second messenger. The first messenger is the neurotransmitter. The second messenger is this yellow molecule, whatever it happens to be. It gets activated, it travels to a nearby ion channel, binds to it, and opens it up, and we get ion flow. There is a tremendous variety of chemical synapses found in the nervous system, both the central nervous system and the peripheral nervous system. And all of those many chemical synapses use a variety of neurotransmitters. Now, within the peripheral nervous system specifically, the most common neurotransmitter in humans is acetylcholine. And the reason acetylcholine is so abundant is because it is the neurotransmitter that's used at the neuromuscular junction. Now, we shorthand acetylcholine like this with a capital A and a lowercase ch. Acetylcholine, if you recall from our discussions of the neuromuscular junction is an excitatory neurotransmitter. And what that means is that when it binds to those receptors that are on the postsynaptic membrane, it stimulates action potentials. Another way of saying that is that the effect of an excitatory neurotransmitter is to generate excitatory postsynaptic potentials, or EPSPs as we call them. When acetylcholine binds, the effect is excitatory postsynaptic potentials or membrane potentials. The membrane will be brought to threshold through the action of sodium rushing through sodium channels into the neuron, and new action potentials will be generated. Acetylcholine, as it turns out, binds to ionotropic receptors. Acetylcholine receptors are ionotropic. Sodium channels open. Sodium rushes into the postsynaptic neuron, and depolarization occurs. Now, any synapse that uses acetylcholine as the neurotransmitter gets a name. We call that a cholinergic synapse. In other parts of the nervous system, there's another really important group of neurotransmitters that are used at chemical synapses. And these neurotransmitters go by the name catecholamines. There are actually several catecholamine type neurotransmitters. For example, dopamine, norepinephrine, and epinephrine are all catecholamine neurotransmitters. Now, these catecholamine chemicals, they're all related to each other. In other words, dopamine is produced in these neurons from certain precursor chemicals, and then the dopamine can be converted into norepinephrine, and the norepinephrine can be converted into epinephrine. So depending on the neuron and depending on which of these neurotransmitters it's going to use, they can be synthesized all from the same precursor molecules. What makes catecholamines particularly interesting is that unlike acetylcholine, the catecholamines can have different effects in different synapses. Their effects can be either excitatory or inhibitory. So remember, we said excitatory neurotransmitters are going to bind to their particular receptors on the postsynaptic membrane and result in excitatory postsynaptic membrane potentials. That means sodium channels are going to open, sodium's going to rush into that postsynaptic cell, and that's going to bring the postsynaptic membrane to threshold so that depolarization can occur and action potentials can be generated. For a catecholamine to have an inhibitory effect, something very different has to occur. The catecholamine has to travel across the synapse, bind to its particular type of receptor on the postsynaptic membrane, and instead of sodium channels opening up, potassium channels will open. Now remember, when a potassium channel opens, potassium doesn't rush into the cell. Potassium rushes out of the cell. The same thing will happen on the postsynaptic neuron. Potassium channels will open and potassium will exit. That's going to make the inside of the cell more negative. That's going to take it farther away from the threshold potential. That's going to make it harder to generate an action potential in that postsynaptic cell, that postsynaptic neuron. So we are actually inhibiting the generation of action potentials. That is the creation of IPSPs, inhibitory postsynaptic potentials. Now, synapses that make use of these catecholamines and their complex effects also get a name. These synapses are referred to as adrenergic. More than 100 different kinds of neurotransmitters have been discovered so far. And believe it or not, we are finding new neurotransmitters all the time. We break neurotransmitters down into different groups based on their structural chemistry. So again, we know acetylcholine is sort of its own unique neurotransmitter. We know catecholamines include dopamine, norepinephrine, and epinephrine. There's a large group of neurotransmitters that come from amino acids and derivatives of amino acids. For example, you may have heard of GABA or serotonin or histamine or glutamate. Some of these are actual amino acids, and some of them are derived from amino acids. And finally, there's another group called neuropeptide neurotransmitters. These are kind of fascinating. These are not as well understood as some of the others. The neuropeptides includes the enkephalin group. Enkephalins are an inhibitory type of neurotransmitter that help human beings and other animals deal with severe stress during shock. I always use the example of a person who is badly burned, very seriously burned. First responders who encounter these people will tell you that sometimes the person doesn't even seem bothered. They're not yelling in pain, they're not crying out, they're perfectly normal appearing, even though they have suffered sometimes third-degree burns over a large portion of their body. And we believe that part of the reason that they're able to maintain their composure in that kind of a situation is because they are flooded with enkephalins. And enkephalins are inhibiting all of the sort of responses that you would expect from a person who has been so badly burned. Now, another thing that we need to have a solid understanding of is the fact that while we often talk about one cell communicating with one cell when we learn about the nervous system, in reality, there are very few one-to-one interactions. In other words, we don't see one axon communicating with one dendrite or one other postsynaptic cell. Instead, we see lots and lots of synapses on each cell. That's what this slide is showing us. On the left-hand side, we're looking at the cell body of a neuron, and we're looking at all of the many dendrites that are extending out from that nerve cell body and branching. We can see the axon hillock right here, and then the first part of the axon, and this is a myelinated axon because we can see some myelin deposited right here. What I want you to notice are all the little dots. Each little dot represents a synapse. Some of the dots are red and some of the dots are green. Everywhere that you see a green dot, you're looking at an excitatory chemical synapse. Everywhere you see a red dot, you're looking at an inhibitory chemical synapse. Different neurotransmitters, different types of chemical synapses. This single neuron is receiving all kinds of information Remember when we talked about different types of neurons and we said the most common type of neuron, 90% of them, are interneurons. They connect one neuron with another neuron. And we said that one of the things that interneurons can do is integrate information. They're going to take all of this incoming information, all of the positive, excitatory, negative, inhibitory information, and they're going to integrate it. They're going to sum it. And they're going to send that summation down the axon to their own terminal branches and send it along to the next neuron. This happens everywhere in the nervous system. Lots and lots and lots of information is being shared all the time. It's not a one-to-one proposition. Take a look at this image over on the right-hand side. This is an actual image taken with an electron microscope. You're looking at a postsynaptic cell here, and this happens to be the cell body. This is a postsynaptic neuron. Notice all of these things. These are all axon terminal branches coming in, widening into a knob, and synapsing with this postsynaptic neuron. So in other words, this neuron that is receiving so much input and is integrating all of it and sending it down its axon. Its axon might end right here in this terminal branch. But look at all the other neurons that are communicating with this postsynaptic cell. The presynaptic cell has to integrate a lot of information and send that information to the postsynaptic cell. But there's not one presynaptic neuron. There are many presynaptic neurons. This is a very, very complex system of input and output. The last thing we're going to be talking about today is going to bring us to a broader anatomical location. We're going to leave the individual neurons and we're going to think about nerves and nerve regeneration. Remember that we said earlier that neurons don't divide once they are mature and in place in the human body. So if a neuron dies, it's gone. You don't see cell division replacing that individual neuron. But neurons can regenerate their axons under certain circumstances. We need the cell body, the neuron cell body, to be intact. And you need at least a little bit of the neurolemma of the axon hillock to be intact. In other words, you need a little bit of the membrane of the axon hillock, and sometimes even down into the axon itself. That's fine too. But you must have at least the axon hillock membrane intact. If the nerve cell body and the axon hillock membrane is intact, the axon can be regrown, regenerated. And since axons can regenerate, that means nerves can regenerate. But, and this is critical, only in the peripheral nervous system. Now, this process of nerve regeneration is not completely understood. There are instances in medicine where we would predict that nerves regenerate, and they don't. And then there are other instances where we would predict that they won't regenerate, and they do. So it's not completely understood, but we do have a general understanding of the steps involved in the process. So remember, we're talking only about the peripheral nervous system. Imagine that there's an injury to your arm, and a nerve inside your arm is transected. It is cut in half. The nerve that is distal to the injury, so whatever piece of axons is on the distal side, that cannot regenerate because it's not attached to the nerve cell body. So that is just going to degenerate. The Schwann cells that may or may not be present on that distal piece of the nerve are also going to degenerate if that nerve happens to have myelinated axons in it. Once that degeneration process is done, all the bits and pieces, the leftover material, will be cleaned up by the immune system. Macrophages inside of the immune system will come and clean up all that debris. The end of the cut nerve that has the nerve cell body attached to it, the neuron cell body attached to it, that's going to undergo a change. And the most noticeable thing that first happens is it swells up. Now, it's likely that the reason it swells is because it becomes suddenly very metabolically active. So remember, you have to at least have the nerve cell body, and you need to at least have a little piece of the axon hillock membrane. The stump that's up near the cell body, the stump of the axon, is going to undergo an amazing transformation. It's going to sprout multiple membrane extensions, multiple membrane processes at the beginning of this regeneration process. Up near the site of the cut, again, at the stump of the axon, the Schwann cells that are left over, if they're present, and the nerve, the axon membrane, are going to form a structure called a regeneration tube. And it literally is a tube, like the tube inside a roll of paper towel. The Schwann cells are going to start secreting a growth promoting mix of chemical signals, including chemicals that are called nerve growth factors. The sprouted processes, these nerve membrane extensions, are going to be directed into that regeneration tube. They're going to be sort of stuck in to the regeneration tube, and they're going to start to grow, and they grow fast. They grow at about 3 to 5 millimeters per day. I know that doesn't seem like a lot, but on a microscopic level, that's pretty amazing. The regeneration tube structure is going to guide these newly growing sprouted processes back to the original location of the axon. In other words, they're going to direct them where the rest of the axon used to be. That's going to help reestablish synaptic contact with other cells. Now, once those new sprouted processes have fully grown out and replaced the missing part of the axon, and once new synapses are established, the process is finished and the nerve cell body is going to shrink back down to its original size. Now, a couple other things to know, and this is important. When a nerve is transected like this, and the distal part of the axon dies, remember that that axon synapsed with something like, say, a muscle fiber. When the axon, the distal part of the axon degenerates and degrades, and the synapse with the muscle is lost, that's a really important loss for that muscle tissue. Muscle tissue needs tone. It needs constant stimulation to be healthy. And what will happen if the nerve supply is disrupted like this? The muscle will start to shrink. It'll start to what we call atrophy without this normal amount of nerve stimulation, without this normal tone. This is something that happens very quickly when nerves are damaged this way. And we give it a name. We call it neurogenic atrophy. The other thing to know is that sometimes nerve regeneration goes wrong. It actually goes wrong quite a bit. Again, if you think about motor nerves that are coming down from the central nervous system and contacting motor effector cells, sometimes during nerve regeneration, the sprouted processes are connected to the wrong effector cell, maybe the wrong muscle cell. Sometimes they never reconnect at all. In other words, the regeneration tube is not able to get these sprouts back where they used to be. So no synapse is reformed. Nerve injury, even when it is repaired, even when the nerve regenerates and you get new synapses formed, it's usually followed by some degree of dysfunction. So even when nerves are regenerated perfectly, they never seem to work quite as well as they did before the injury. And of course, the other thing to remember is this. Nerves that are damaged in the central nervous system, the spinal cord and the brain, do not regenerate. One of the things that we talk about when we learn about the vertebral column and the skull is how important those bony structures are for protecting the central nervous tissue. The central nervous system can't repair itself, so it's critically important that the central nervous system tissue be protected as much as possible. That is the job of the skull around the brain. That is the job of the vertebral column around the spinal cord. On this last slide, we just have a diagram of nerve regeneration. I like this diagram because it really lets us think about these sprouted processes at the end of the proximal axon stump. Up at the top, we're looking at what we would call a multipolar neuron. Here's the cell body with some dendrites coming off. Here's an axon, it's myelinated. Here are the terminal branches, and here's normal muscle. Now, the red arrow here indicates an injury to this nerve. Remember, the nerve is not going to have one axon in it. The nerve is going to have many, many axons in it. The ganglion is not going to have one nerve cell body in it. It's going to have many, many nerve cell bodies in it. But for ease of understanding, they've drawn it like this. So here comes the injury. We have disrupted the nerve in the midst of the axon here. Now, about 2 weeks after that initial injury, you're going to start to see regeneration. The nerve cell body, the ganglion, in other words, is going to swell. And this proximal end of the stump is where we're going to see regeneration occurring. This distal end, from distal to the injury, is going to degenerate. They're showing us macrophages moving into the area, and they're showing the myelin being taken apart. You can see the axon is now a dotted line. It's being destroyed and it's being removed from the area. Remember, you're going to see at the end of the stump all these little sprouts produced. Now, you can't see a regeneration tube here, but remember, all of these little sprouts are going to be redirected into the tube. They're showing us all of these Schwann cells here. It's the Schwann cells that are going to create this tube. And all of these little sprouts will be directed into the tube so that they can be then directed to the location where the new axon needs to go. And of course, we need that location to be back to this particular muscle. And we need new synapses built back to this particular muscle. Now, months and months after the injury, you wouldn't know by looking at it that that nerve had been transected, but it's unlikely that it will function exactly as well as it did before the injury. And of course, sometimes we don't see the result we want. Sometimes either we get a connection to a wrong effector cell, or we end up with a connection that just can't make it. So you can see all of these sprouted axon processes here, but they never connected. They never made the connection back to the effector cell. Nerve regeneration is critically important in the peripheral nervous system, but it is not perfect, and it is not present at all in the central nervous system.